Bismillahirrahmanirrahim. Assalamu alaikum. Welcome to Biosciences with Shadrin Sawati, and I'm here to deliver the lecture from my next series, which is biotechnology series. And as a biotechnologist, I will try my best to clear all your concepts related to the biotechnology because I got lots of questions related to the basic concepts of the and terms and conditions of the biotechnology. So I decided to deliver the lectures from the biotechnology. I will discuss each and everything related to the biotechnology. So must watch all my lecture. Do subscribe my channel, like my videos, and share my videos. If you are sharing my videos, you are sharing the knowledge. So keep watching, keep sharing knowledge with others, and kindly do viral the education and educational content. So today from the biotechnology, I will deliver my first lecture about the DNA sequencing, and I have discussed in detail about the DNA, genes, and genetics in my genetics series. So must watch all my lectures from the genetics series. You will get a lot, and the basic concept about the genes and genetics will be clear upon you people, and this lecture will be easily understand for you people. So DNA sequencing means the to know the actual DNA sequence of any person, any species, from plants, animals, human being, from anything else. So what is DNA sequencing, and what is the basic concept of the DNA sequencing? I will give you here. So I will discuss main principle of the DNA sequencing. Then I will discuss the Sanger method. Especially here in this lecture, I will I will start the Sanger method. Then my next lecture I will deliver the Maxwell-Gilbert method, and then the next generation sequencing. So I discussed the history of the Sanger method. Then I will give you the some basic concept of the DNA structure and the DNA replication, and also deoxyribonucleotide and the deoxyribonucleotide concept, and the main steps of the Sanger method, which I will discuss in my upcoming lecture. Okay, so the first of all, the basic concept or basic principle of the DNA sequence. What is that? To generate pieces of the DNA, to generate pieces of DNA of different sizes, all starting from the same point and ending at the different point. Number two, separation of these different size pieces of the DNA by gel electrophoresis. Gel electrophoresis is used, maybe you are familiar with the gel electrophoresis that is used for the separation of the charged particle depending on their size, depending on their charge, like proteins, DNA, RNA, and so on. And reading of the sequence from the gel. All sequencing method, whether it is the Sanger method, it is Maxwell-Gilbert method, all uses this basic principle. Okay. So before discussing the Sanger method, I will discuss in detail. I will discuss something related to the history of the Sanger method and the basic concept about the DNA and replication and PCR. So what is Sanger method? Sanger sequence, also known as the chain termination method. Why it is called chain termination method? I will show you and explain. Is a method for determining the nucleotide sequence of the DNA. The method was developed by the two-time Nobel laureate Frederick Sanger and his colleague in 1977. Hence the name Sanger sequence. In reward to the invention, the method of the sequence was named as the Sanger sequence. And here you can see the Frederick Sanger. I added the photograph of the Sanger, to tribute them and to reward them, reward him for his such a tremendous achievement in the biotechnology, in the science and technology. So Sanger, born in 13th August 1918, and then died in 19 November 2013, was a British biochemist who twice won the Nobel Prize. Sanger twice won the Nobel Prize in chemistry, one of only two people to have done so in the same category. The other one is the John Biden in the physics, who got the Nobel Prize 2 times. Okay. Now, what is the DNA? The structure of the DNA, which is the deoxyribonucleic acid. You are familiar with the DNA structure. DNA is the double-stranded, and the DNA consists of polynucleotide, two polynucleotide chains, which are helix-like structure, and these two strands are complementary with each other, and each strand consists of the deoxyribonucleotide are nucleotide simply, and the single nucleotide, as you can see here, each nucleotide consists of the phosphate group, the nitrogenous base, and the deoxyribose sugar. Is it clear? So one nucleotide consists of how many groups? 3 groups. And this nucleotide consists of 3 groups, and it may be consists of the 2 groups, nucleoside plus phosphate group. Now, what is nucleoside and nucleotide? If you have to differentiate, nucleotide consists of the 3 groups, and nucleoside consists of just 2 groups. Ribose sugar, deoxyribose plus a nitrogenous base. In nucleoside, just 2 deoxyribose sugar plus nitrogenous base is present. But in the nucleotide, phosphate group is attached to the nucleoside. So this is the clear difference between the nucleotide and nucleoside. And you can see here there are 4 types of the nucleotide present in the DNA: adenine nucleotide, guanine nucleotide, and cytosine nucleotide, thymine nucleotide. As we know that they are all complementary. Adenine bond with the thymine, and cytosine bond with the guanine, means A with T and C with G. You are familiar with this concept. Now I'm here not discussing in detail, just giving you the concept about the basic structure of the DNA. Okay. Now a little bit about the DNA replication. As we are familiar that the DNA replicate in our cells during the interphase of the life cycle of the cell, in which this double-stranded DNA first, you can see here, become the single-stranded with the help of the enzymes present inside the cell, present inside the nucleus of the cell. So double-stranded converted into single-stranded, and along the single strand, new strand is synthesized. As you can see here, the blue and the yellow strand is the new or the daughter strand, which synthesize along the parent strand. So the synthesis of the DNA occur from 5 to 3 end, must keep it in human. And I will discuss in detail. Here I'm not giving the concept of the 5 to 3 end and 3 to 5 end. So just here mentioning the DNA replication, how it will occur in the cell, or how it will be helpful in understanding the concept of the Sanger method and the PCR. So this is what the DNA, which is double stranded, first is converted into single strand in order to form the 2 copies or the replica of its of DNA. Okay. Now, the polymerase chain reaction. This polymerase chain reaction, which is polymerase chain reaction, PCR. This is used for forming the, you can say, the clones or the copies of the gene or DNA. So here you can see the PCR consists of basically 3 steps. I will not discuss in detail, but later I am giving you the concept. That first one is that denaturation. Denaturation is that step in which the double-stranded, you can see here, this is a double-stranded DNA. This double-stranded DNA is converted into single strand, because here the copies of DNA is formed. Copy of DNA or gene is formed. So like the replication, like the simple replication in the nucleus of the cell, what will happen in the PCR? Here in the nucleus of the cell, there are many enzymes which help in converting the double strand into single. But in case of the PCR, we use the high temperature to denature the double stranded into single stranded. So the second step is the annealing. In annealing, we need the primers. Primers are the small nucleotide sequences, and these primers help in annealing. Means during annealing process, what will happen? The primers attach to the single stranded DNA, which is formed by the denaturation, and this primer attached and guide or signal to the nucleotides present in the PCR mixture, and then these nucleotide, which are the deoxy nucleotide, move towards the single stranded DNA and form the double stranded DNA, and then the last step is the elongation, in which the single stranded DNA is completely converted into the double stranded. So denaturation, annealing, and at last the elongation, and all these steps require different temperature, and I will discuss in detail in my lecture about the PCR. Okay, the concept clear? So this concept is used in the Sanger method. I will show you. So must clear all these things before to understand the clear concept of the Sanger method. So now I'm going to discuss what are the dideoxyribonucleotides. Dideoxy. First of all, you should know what is the dideoxy. Di means two. Deoxy means D means removal of oxygen from the ribose sugar. Is it clear? So dideoxyribonucleotide. There are basically two types of ribonucleotides. Oxyribonucleotide, which is present in the RNA, and deoxyribonucleotide, which is present in the DNA. The difference between the deoxy and oxyribonucleotide is the presence and absence of the oxygen atom at the carbon number 2. This is the ribose sugar. Ribose sugar, you people are familiar that this is 5 carbon sugar. It consists of 1, 2, 3, 4 and 5 carbon. So what will happen in case of the oxyribose, means in case of the RNA, here hydroxyl group is present. But in case of the DNA, which is deoxyribonucleotide, in case of the deoxyribonucleotide, hydroxyl group is removed, or you can say the oxygen is removed here and just hydrogen atom is present at carbon number 2. Is it clear? Now, what will happen in the dideoxyribonucleotide? In case of the you can here clearly see the difference between the deoxy and dideoxy. In dideoxy, what will happen at carbon number 2 and carbon number 3, just hydrogen atoms present. Hydroxyl group, or you can say the oxygen is removed. So what will happen? Why the Sanger used the dideoxy? The reason is that the oxygen which is present at the carbon number 3 of the ribose sugar help in the formation of the phosphodiester bond or phosphodiester linkage between the nucleotides to produce the to form the DNA polynucleotide. So this oxygen atom which is present at the carbon number 3 bond with the other nucleotide at carbon number 5, and the bond is known as phosphodiester bond or phosphodiester linkage, which is formed between the carbon number 2 of one nucleotide and carbon number 5 of the other nucleotide in the polynucleotide chain or DNA. But what will happen in the dideoxy? Why the Sanger used the dideoxy? The reason is that the oxygen here is removed. So this dideoxy stop, or you can say that terminate the chain, because oxygen is not present here, and this, if the oxygen is present, then this will form the phosphodiester linkage between the nucleotides. So here oxygen is absent, so what will happen? The chain terminate, or you can say the DNA synthesis stop. DNA synthesis stop if the dideoxyribonucleotide attached to the single stranded DNA during the DNA replication or DNA synthesis. Is it clear? If this dideoxy attached here, I'm showing you like this. This you can see here, this is the single stranded DNA, and you know that the during replication, whatever the second daughter strand will produce along this parent strand from 5 to 3 end, with the addition of the deoxyribonucleotide and nucleotides, then what will happen when the dideoxy attached at any point along the single stranded DNA, then it will stop or terminate the formation of the what? DNA. Is it clear? So that's why Sanger used the dideoxyribonucleotide, and the reaction is known as the chain termination reaction. Is it clear? So you can read here the deoxyribonucleotides are chain elongating inhibitors. They are inhibitors. They inhibit the DNA polymerase to synthesize the DNA. Is it clear? Used in the Sanger method for DNA sequences. They are also known as 2,3 because both the 2 and 3 position on the ribose lack hydroxyl group. 2 and 3 position lack the hydroxyl group. But in case of deoxy, hydroxyl is, and in case of the oxy, both 2 and 3 hydroxyl group present. Now there are 4 types of the nucleotide. So there are 4 types of the dideoxyribonucleotide. Like ddNTPs are ddGTP, maybe ddATP, ddTTP, ddCTP. So there are how many types of the dideoxyribonucleotide? 4. Like the deoxyribonucleotide, there are 4 types in the DNA. So there are 4 types of the dideoxyribonucleotide, which are used by the Sanger for the chain termination, which I will discuss later on. So ddGTP, ddATP, ddTTP, ddCTP. These are the 4 dideoxynucleotide. Okay. How sequencing is done? As I have discussed that Sanger used the dideoxynucleotides, and these dideoxynucleotides, whenever attached at any point of the single stranded DNA during the elongation process, during the formation, or you can say that simply you can say that during the formation of the DNA, double stranded DNA, what will happen? Whenever the dideoxy attached, it will stop the formation of the double stranded DNA. Simple, in simple words. As the nucleotides are complementary, so ddNTPs are also complementary. And whenever they are attached with like G DDP attached with the C nucleotide of the parental strand, then it will stop the elongation of the chain or the formation of the DNA. So this mechanism was used by the Sanger to stop and to read the sequence of the unknown DNA. Okay. Now I will discuss in detail about the main steps to the Sanger sequencing. But before discussing the Sanger method, I will share something about the required material we need for the Sanger method or the Sanger sequencing. A single stranded DNA template must keep it in your, which is unknown sequence. This single stranded DNA are the DNA of unknown sequence. Number 2, the DNA primers. Number 3, the DNA polymerase, which help in the, and DNA polymerase here is the tag polymerase. You are familiar with that, which help in the formation of the some double stranded DNA. And normal deoxynucleotides, triphosphate, means the ddNTPs. They are represented as ddNTPs. ddNTPs are normal nucleotides. Must keep it in your mind. ddNTPs are normal and ddNTPs are dideoxynucleotides. Simply you use the word like deoxynucleotide and dideoxynucleotides. So the deoxynucleotides are DNTPs, and if they are dideoxy, they are known as ddNTP. And ddNTP help in the termination after DNA strand elongation. Is it clear? Means stop the synthesis of the DNA. Is it clear? So I will discuss in detail about the Sanger sequencing process in my next lecture. So must watch my all my lectures related to the DNA sequencing, and I will be back tomorrow. Till then, Allah Hafiz.